and they have to move, they have to sense, and so on. Yeah, big old pons here. Your pons is quite large compared to the human situation here. Your brainstem, little bump right here. Probably this is cortical spinal fibers coming down through. Cerebellum, pretty obvious here from this side as well. Maybe we could even see the insular cortex if we pull this open a little bit. So I'm pulling aside the temporal lobe from the parietal lobe. This dolphin brain is a little bit looser here. It's older probably. See if we can see that insular. You can kind of see, I think, I don't know if you guys can, but I can kind of see a little bit of cortex way deep down at the bottom of that that sulcus there. That's going to be your insular cortex in the dolphin. So there you go. Bet that's the first insular cortex you've seen today in a dolphin. See if we can see anything in the midline here. You got your cingulate gyrus is gonna kind of go along the top of the corpus callosum, which we're looking at from the top here. It's kind of hard for me to identify exactly where it is. I don't know the neuroanatomy of the dolphin very well, so it's kind of hard for me to say exactly where it is, but I would assume that the position is right there hugging the corpus callosum as well. Another thing we can kind of see, this I believe is going to be your colliculus. That's your superior colliculus. So that's going to be a visual relay center. This is the kind of thing where, like, I throw a ball at your face, you reflexively pull up your hand. Your colliculus is going to do a lot of that visual processing instead of sending that whole message back to the occipital cortex. The dolphin will, like, sort of, or you or dolphin will identify, like, oh, that, you know, Steve is punching me in the face or something. That kind of information can go a little bit, takes a little bit longer, goes a little bit slower. You really need to react and kind of block my punch. Initially, that's going to be the kind of thing that's dependent on your visual, your superior colliculus. It's real big in a dolphin. I would guess that that has functional consequences. Perhaps they have some, you know, hunting fish and whatnot is going to probably use really very rapid visual information. So my guess is that that is why that is the case. Not a whole lot else we can really see here in the dolphin brain. Okay, let's move on. Here is a rat brain, slightly smaller than your dolphin or your human. A lot of other things that are quite different is you don't have all those cortical folds. Here's the top of the brain, the cortex. Cerebellum's real big. Trying to get a good view here. Okay, cerebellum's real big. You got your brainstem and spinal cord coming off the back. Here are the olfactory bulbs. They're quite large. Rats do a lot of smelling. Big olfactory bulbs sticking off the whole front of the brain there. Let's look at it from the bottom. A couple other things we can see. Here is your pons, and then the beginning of the spinal cord. That little circle kind of thing right there is your bottom of your hypothalamus. That would be kind of where the pituitary comes out I was showing you. The little white bit right there is your optic chiasm. I don't know how well you can see that with this camera, but that's what that is. It's a little bit kind of screwed up probably from removal or whatever, but that's what we're looking at there. A lot of the similar structures, same type of deal. Obviously the proportions of stuff are a lot bigger, different than in these other higher sort of mammal brains, but basic organization is going to be the same. Okay, let's look at a couple other brains. So here we got a turkey brain. This is a bird brain now, our first bird brain. Couple things we can see here. Big cerebellum. It's a little bit torn up, but that's what it is right there. Here's your forebrain. And then these, like, sort of giant, this right here and this right here are your superior colliculus. So again, you can see they're almost as big as the whole forebrain, or half as big as the whole forebrain. So a lot of rapid visual processing would be, I think, the functional implication of that. A bunch of other things are sort of organized differently in the bird brain. I'm not a super expert on bird brains. If you're ever interested in this, you can go talk to Dr. George Streeter. He's really one of the world's expert on bird brains, and other comparative neuroanatomy types of things, by the way. Here, just FYI, is your pons, your spinal cord coming off, bottom of your colliculi, huge colliculi. Optic tracts are coming here. I would assume a lot of this, you know, bird, turkey, etc., is going to give a lot of visual components to its sort of world as well. So I assume that this occipital cortex here is going to be doing some of that, or whatever the analogy, I'm not sure what it's called in a bird brain, probably something slightly different, but it's also going to be a play a large function in the sensory world of this animal. Okay, another thing, here's a squid brain. So this squid brain is not real big. These are mostly eyes you're looking at. I've left the eyes attached since it's rather difficult to remove them from the squid brain. One thing that's kind of interesting about the squid brain, is also true of the octopus brain in general in cephalopods, is that their brain is essentially kind of a ring-shaped structure, and it circles their essentially esophagus. So their mouth, the mouth of the squid would have been right about here. Food, when it eats, would go down right through this hole. That is surrounded by brain. So if we kind of try to pull it away here, I've left a little bit of it here. You can kind of see that ring. I've tried to preserve the brain and remove the rest, so you kind of see that ring type structure. I don't know how easy it is to see here, but that's basically your brain of your squid. These things aren't too smart. The brain's kind of small. Octopus would be bigger proportionally. I don't have a large octopus, so I don't have a good dissection of one, but that's kind of what we're looking at. And as you know, the octopus brain, I think I mentioned, is also got a unique feature of that it's sort of distributed. So it has the central ganglion that are attached to the eyes, do a lot of the visual processing, probably do the integration of all kinds of information, but they have these other sort of mini brains in each of their arms that do a lot of sensory and motor processing. So they can, for example, kind of detect what is the surface that I'm on and then create camouflage that looks just like that kind of thing. Also, it can, like, you know, move each of the individual suckers and the arm itself in very complex ways, and all that takes a lot of computation, and a lot of that's done out in the periphery, out at the arms themselves, like a little mini brain for each arm almost. Pretty cool stuff. Okay, here is a shark brain. Here's the eye. So this shark was probably, I don't know, probably about as long as this tray here. Maybe even a little bit longer. Brain pretty small, eyes pretty big, brain pretty small. Here's what we got, mostly spinal cord. Looking at it from the top, it's a little bit torn up. It's kind of hard to pull a shark brain out. They don't really have bone. It's like they have cartilage. It's difficult dissection. I did my best. But the main things to point out here are huge olfactory bulbs. They're mostly torn up, but a lot of olfactory bulbs. These things are smelling blood in the water and so on. That's a big part of that processing. Forebrain, pretty much tiny. This is going to be, I think, part of the forebrain as well, but basically the forebrain's pretty small. This guy is made out of brainstem. That's what we're talking about here. These things are pretty primitive creatures. They have a lot of reflexive responses, a lot of kind of stimulus response sort of thing. See fish, hunt fish. They're not planning. They're not doing a lot of social interactions, etc. They're a mostly brainstem-oriented creature. And I'll leave that at that. I have, let's see, moving on. Next thing I'm going to show you is, actually, let me just put my gloves off here. Okay, so now we've got some slides. So these are prepared brains. What we're looking at here is a chimp brain. The big one is a chimp brain. I've also got a rat brain sections right underneath it, so you can see, number one, the size difference, number two, some of the same structures. I don't know how well this is going to work with this video camera, but I just want to point out a few things since I got this brain here to look at. Let me grab a pointer. Okay, so a couple things here. So we're up in the front of the chimp brain here. What you can see, first of all, is your caudate, putamen. You got your internal capsule here separating them. White matter tract separating caudate from putamen. You got cortex. So you have your cingulate cortex here. Here's your corpus callosum. We're looking, by the way, at a coronal cut. So this would be something like a cut like this. Let's see. White matter is the stain here. Am I still going? Yes. Okay, so white matter has been stained black. Gray matter is white on this particular preparation. Here's the rat analogy. I don't know how well you can see it, but you're up here in the prefrontal cortex. Here's at the very beginning of this striatum. First thing, this white matter tract is analogous to this white matter tract. So here it's black, here it's more like white. But those are the same, basically connect to the corpus callosum. These are all white matter fibers that are coming from cortex off to other places, either other cortex or down to, you know, the spinal cord, etc. Here is your striatum. You don't have a separation of the caudate and putamen in the rat or mouse. So it's just one big mass. It would be as if this whole thing were connected instead of having that white matter tract in the middle. Here's another commissure. It's called the anterior commissure. It's traveling from front to back at this portion of the brain. Kind of hard to see it here on your chimp brain. We're a little bit further back here. Now we're starting to see that white matter tract. Your caudate's mostly gone. You got your tail. Maybe this is the I may be screwing this up. I apologize. Let's see, corpus callosum. You got a little bit of septum, I think this is here. That's about all I'll say about that. Let's move a little bit further back. One thing I wanted to show you on this one is that here's your anterior commissure. It's connecting in the middle. It's connecting the two hemispheres. So here you have the corpus callosum and also the anterior commissure. This commissure, I'm not sure where it's coming through here. This might be it, actually. Some of these fibers travel from front to back, so you can think of it as coming towards you. And then they cross. And that's what's happening here. So they're starting to cross here. Here's where they actually cross. So it's sending information usually from subcortical structures like striatum, globus pallidus, over to the other hemisphere. All cortex on the outside, of course. Another thing to show you here, I just wanted to show you this one because it has a nice view of the globus pallidus. So you have your external and internal segments of the globus pallidus. This is something we talked about in the basal ganglia lecture a bit. Here's your striatum. Here's part of thalamus, and we're starting to see the beginning of what's called the epithalamus or the habenula. So this is the, probably there, maybe this might even be thalamus still, but we're right almost getting to where the habenula is. Here you have your habenula as well. It's sitting right up on top of the thalamus, super evolutionarily ancient. That's why it's so easy to find, more or less, across species. It's really kind of separated from thalamus, but right above it in pretty much all these species. Plays obviously an important role. Another thing to show you on this one is over here in the lateral parts, now we have all this cortex, it's gyrus and sulci and everything, white matter tracts. But you also have this little mass of tissue here. This is going to be your amygdala. This is gray matter. This is mostly basal amygdala in a chimp, like we're looking at here. Over here, we're a little bit further back. Now we're behind the amygdala. Now we're starting to get into the hippocampus. You can already start to see, probably this is like the transition right here. Hippocampus is just starting. Here you can really start to see those layers of the hippocampus and your dentate gyrus and your CA regions. Here's another one a little bit further back. Again, really nice jelly roll type structure of the hippocampus, very visible. Another thing visible here is the start of the substantia nigra. Even easier to see here. Here's your nigra, here's your white matter tracts that are just underneath